is Pharma Forum web editor Nicole Raleigh, and I am on site at ESMO 2024 in Barcelona, and sat next to me today is Israel, or Izzy, Lowy, who is the Senior Vice President of Clinical Oncology at Regeneron. Pleasure to meet with you.
Thank you very much. It's a pleasure to be here.
So if we skip an event back to WCLC the other week, there was 5-year survival data presented there on the PD-1 inhibitor Libtayo as a first-line monotherapy in advanced non-small cell lung cancer, or NSCLC. But here at ESMO, longer-term results with investigational fianlimab, a LAG3 inhibitor, plus Libtayo, in an initial trial in advanced melanoma has been presented, and it has shown high clinical activity. So the reason I've got you here with me, Izzy, is I want you to tell our viewers more about that data and what it means for not only the space, but also for the patients there in that space.
Sure. So we're really happy about all of this data, as well as other data that we're developing. So first, on the lung cancer data, we first read out this trial 5 years ago on an interim analysis, and it was positive, and it led to the approval of cemiplimab, known as Libtayo, for patients with advanced lung cancer, both squamous and non-squamous. We're one of very few, actually I think only 2, anti-PD-1s that has approval as either monotherapy or in combination with chemotherapy for both the non-squamous and squamous subtypes of advanced non-small cell lung cancer. So what we presented at the World Lung Cancer Conference was 5-year follow-up survival from our initial readout, and we were thrilled to see the durability of the benefit that patients had with a median of a 26-month survival compared to 13 months for the chemotherapy control arm. And at 5 years, we basically doubled the number of patients that were still alive at 5 years, from 13% to nearly 26%. We also, in that study, showed some preliminary data suggesting that if patients were started on monotherapy, that if you, at the time of progression, if you added chemotherapy and kept the Libtayo going on, that also provided additional benefit superior to what it would have been if someone had started out on chemotherapy alone. So although this was not a randomized study and it's not part of our label, we thought it was a novel aspect of our study to show that we also can give guidance to clinicians and patients who are struggling. Should they start on monotherapy, or should they start on treatment with chemotherapy from the beginning? That if they're candidates for monotherapy, they can benefit from waiting and seeing how long they do on the monotherapy. What we presented here at ESMO is work that we're doing in melanoma. We started a first-in-human study also quite a few years ago, where we treated various different patients with different tumor indications with the combination of our LAG3 antibody fianlimab in combination with cemiplimab. And what we've done is we were struck initially by a very high response rate, higher than what others had been seeing with their competing combinations. And so what we did was we enrolled several cohorts. And so we have 3 independent cohorts totaling nearly 100 patients. And we maintained this high response rate of nearly 60% with a fair amount of complete responses. What we've been able to do with this long-term follow-up is demonstrate that the responses are quite durable. So we've lost barely any of them. And patients who initially responded with what's called a partial response, over time, quite a few of them evolved into complete responses. So initially we had complete responses in the range of like 12 to 15%, but now with this additional follow-up, we have complete response rate of nearly 25%. Moreover, because we have this additional follow-up, we have seen a progression-free survival where our initial estimates were around 15 months, and now it's actually looking more like 24 months. So this kind of accumulating data that we've shared, and a key aspect also of what we presented was the response analysis was done by independent reviewers. So this wasn't investigator response assessment, which can be biased at times, and that's why you have independent reviewers. So we are very confident in the responses in this group of patients. We are pleased with the durability. We are pleased with the evolution of complete responses. We are pleased also that we saw activity in patients who might have risk factors that make you think they would be less likely to respond. High LDH, liver metastases, and also in one of the cohorts, we allowed patients who had treatment with PD-1 during an adjuvant earlier phase and who after at least 6 months after completion of that adjuvant treatment had a relapse. We showed that we could still get them to respond well. So this has allowed us to have the confidence to mount a rather broad phase 3 program in melanoma. We have a phase 3 in advanced melanoma that will read out next summer. We have a head-to-head against the only other approved LAG-3 PD-1 combination because we're confident that we can show that it's a better treatment effect. And we have also developed registration studies in the adjuvant setting and also in what's called the perioperative setting, where patients are going to have surgery. They get neoadjuvant before surgery and followed up afterwards. And on top of that, we are doing some exploratory studies, the most important one being in lung cancer, where we have several phase 2 studies underway. One where we are combining cemiplimab and fianlimab versus cemiplimab, and then another for patients with high levels of PD-L1, and another study that we're doing where we're combining cemiplimab and fianlimab versus cemiplimab all on top of chemotherapy, which is for patients with any level of PD-L1. Both of those are building on our approvals that we have with cemiplimab, Libtayo as a monotherapy or in combination with chemotherapy to see whether we can augment that further. If those trials, which we hope to have preliminary data by the end of the year, if those trials pan out, we would then move those into registration efforts as well. We were pleased to hear, other reports at this meeting that the addition of LAG-3 to other people's LAG-3 to PD-1 can augment the response rate seen in lung cancer or certain, sub-selected groups of that, giving us additional hope that we, given that we have such a strong anti-PD-1 and we believe potentially a best-in-class LAG-3 antibody, that we will see the same. And we have other exploratory cohorts going on as well.
Impressive results indeed.
Thank you.
These words, hope and confidence and being pleased, very much apt by the
Yes.
sound of it. Yes. So if we think about Regeneron's sort of general ethos when it comes to the oncology pipeline, I mean, I've just said impressive results, but overall, what does this potentially benefit the field as a whole and the progression and transformation of oncological research going forward?
So a big focus of our efforts has been to really develop a toolkit of agents that are active in immunotherapy in particular, with the goal of being able to combine these agents in a facile way. So our first step forward was with anti-PD-1 Libtayo. Our next big step is combination with anti-LAG3 fianlimab. We have several other pillars of our approach that engage the immune system. A lot of them are bispecific antibodies. So they either engage T cells through CD3, so they are CD3 directed bispecifics. We've also pioneered the approach to target another key lever in engaging the immune response, that's CD28. We call those co-stimulatory CD28 bispecifics. So we have a panel of these in the clinic. Actually, in hematologic malignancies, we just recently earned an approval for our first CD3 bispecific, CD20 by CD3 for lymphoma, known as now Ordspono, was approved in the EU, and we're waiting for reviews in the US. And we also have a myeloma BCMA by CD3 that is under review in both the FDA and EU. Those are run by my colleague who runs the hematologic malignancies, but we share the mechanisms of how we're approaching cancer. We have a number of CD28 co-stims in the clinic. We have, in the past year or so, we demonstrated proof of concept that this mechanism can work. And we're very excited about that. We are now trying it in multiple other tumor types and learning our way forward to understand the rules for how to best combine these agents that we're studying in patients. We have other mechanisms that we're exploring as well. We have developed a new targeted cytokine approach that will deliver cytokines hopefully directly towards the tumor microenvironment, focus the effect of the cytokines to also augment immune responses. We call this signal 3. We also have recently acquired a company called 2seventy, which we were collaborating with for many years to look at the opportunity of cellular therapies that are sort of, the CAR T cells and TCR T cells, to see whether or not we can help develop best-in-class therapies by virtue of developing a really good cellular therapy and being able to combine it with elements from this toolkit that I just described. So that's now called Regeneron Cell Medicines. It's another division in our company. And we've just initiated a clinical trial there with a CAR T cell targeting ovarian cancer with MUC16. We have a MUC16 by CD3. We have a MUC16 by CD28. We have fianlimab. We have cemiplimab. We have our targeted cytokine. All of these things we think can approach the immune system from different angles to make the best immune response possible. So I think some people are concerned that we've sort of hit our plateau in terms of what checkpoint blockade can do. I don't think that's the case. I think it's really, we've gotten the easy stuff done. Now we got to take it to the next level and figure out intelligent combinations that can really move the field forward.
Fascinating, exciting. And let's bring it back to the conference. Anything, perhaps not, of the same level of excitement and potential that you've heard trends about or takeaways from ESMO 2024? I mean, so much innovation has been discussed at this Congress this year. And, just the Regeneron example is perhaps enough to be exploring. But if there's anything you wanted to share with viewers that next on the agenda for
Well, it's a very busy conference. I think, at this conference and also at World Lung, there was some, potentially exciting data about new constructs combining VEGF blockade and PD-1. That's certainly, it's early days, but, whether or not that might be a significant step forward. As I mentioned earlier, at this meeting, there was a presentation showing that the addition of LAG-3 to PD-1 plus chemo can enhance the basal effect that you see there, which gives us also more confidence that we're on the right track in testing this in lung cancer. I guess it's, I've been busy meeting people and talking to people, so I need time to reflect and look at everything else there. But I think those are two things that I heard about that caught my attention. And I think what's most exciting is the progress that's being made on so many fronts to convert cancer from what used to be a death sentence to something that can, if not cured, at least can be managed chronically and continue to live a quality life.
Exactly. Well, it's been a pleasure, Izzy. Thank you for your time today.
Thank you. My pleasure as well.